Okay, everyone. So this is going to cover section 3.5 on cell growth and division. Now, this is going to be a really base level video because honestly, if you were taking another biology course, cell growth and cell division could honestly be its own section, its own exam, or at least a really good portion of an exam. So I'm just going to take this really basically, okay? So, first of all cells have a life cycle, and there's two phases of that life cycle. There's interphase and there's mitosis. Okay, so it says right here interphase and mitosis. Most of the cell's life is spent in interphase. You can think of it as the in between time when the cell is just doing its normal processes and not dividing. Mitosis is the division of the chromosomes, the genetic material, and everything else that the cell needs to form into two identical, fully functional cells that are exact copies of each other. And then lastly, cytokinesis is when the cytoplasm is cut in half and we finally get two distinctive cells. But let's first talk about interphase. So during interphase, you can look at this life cycle right here. There are distinct phases, G1, S, G2, and M is mitosis, or the actual division part. Look how small of an amount of overall time of a cell's life cycle that the mitosis part actually is. So in this G1 phase, cells are not actively dividing. The cell is kind of just growing, it's carrying out its normal functions, and it's just kind of doing cell things to just make it real simple. We call this the G1 or the gap 1 phase. And it's basically the growth phase. So you could think of it as gap 1, you could think of it as growth phase, um, whatever. There's also this G0 phase that cells will go into after they've divided and don't need to divide anymore. Perhaps they're too old, or perhaps there's certain cells that only divide during when an organism is developing, and then they stop dividing afterwards. But so let's get into that G1 is sort of just cells doing their own cell processes. Okay. For cells that are going to divide again, G1 is followed by S phase. During S phase, which is S stands for synthesis, that's when a cell is going to replicate its DNA. So when Dr. Droud talked about how DNA is replicated, that happens during the S phase. You can think of it as the synthesis phase, where it's going to synthesize, that means to make new copies of its DNA. After synthesis phase, or S phase, it goes into G2, which is, you can call it second gap phase, you can call it second growth phase, but basically, in G2, the cell continues to grow, and it makes necessary preparations for mitosis. So at this point, it's got two copies of its DNA, so it's going to copy other things that it needs, like the mitochondria or other organelles, and just get ready because the next stop is mitosis, okay? Just really quickly, you can read all of this later, but um, thinking about how long this takes, cells are going to spend most of their life in G1 phase. It's here the cell might spend a couple hours or even just many days. In the S phase, how long it takes to copy all of the DNA lasts between 8 to 10 hours, and G2 approximately 5 hours. So think of that like an entire life cycle of a cell sometimes is a matter of hours before it's ready to copy again. Um, in contrast to that, that G0 phase, where it might never have to actively divide again, could be the final resting place. It could just stay the rest of its natural life cycle until it is ready for senescence or cell death. Apoptosis is called cell death. It could just stay in that for the rest of its life. Um, let's talk about now what chromosomes look like. So this gets really confusing for students because there's a lot of terms that have to be thrown around and kept like steady in your brain. So let's go through them real quick, real easy like. Okay. During that S phase, when the chromosomes are condensed, if you remember, when they're not condensed, they're called chromatin, and then they condense into these sort of structures that you're more familiar with, and they're called chromosomes. But during that S phase, they have to copy the amount of DNA, which will double. So you go from one and you double it, that's two. Um, so after that S phase, each cell will actually contain two copies of each chromosome. And each copy of that chromosome is referred to as a sister chromatid, and it's physically bound to the other copy. So take a look here. Let's say that the red one, there's a red and there's a blue. Let's say the red is from dad and the blue is from mom, okay? Because you get a copy from mom and you get a copy from dad. You also have to copy each copy. So notice that there's two reds and there's two blues. So when we talk about the two copies of the red from dad, let's say, and the blue is from mom, I think that's what I said, but let's say red is from dad, blue is from mom. When we talk about the two red copies, we call those sister chromatids. But when we talk about the red copy and the blue copy, we call those homologous chromosomes, okay? So homologous chromosomes are when we talk about a copy from mom and a copy from dad. When we talk about the copy of mom and dad, so the copy of dad and the copy of mom, we call those sister chromatids. Okay. Sister chromatids are physically bound to each other by this structure called the centromere. So it's just this little structure that holds sister chromatids together. There's another structure called the kinetochore that will hold the duplicated homologous chromosomes together. That's the kinetochore. And they're sort of in like the same area, right in the middle. But once again, centromeres hold sister chromatids together. The kinetochore holds homologous chromosomes together. And so once again, all of this is really just going to be a basic walkthrough because we could spend a 50-minute lecture talking about each step of mitosis here. But let's just do really basically, okay? So now we're in M phase. Look at this. M phase typically only takes 1 to 2 hours. And during this phase, a cell undergoes two major processes. First, it completes mitosis, and then it does cytokinesis, where it just divides into two. So let's talk about the phases just really basically. So in prophase, what you'll see is the chromosomes will condense. Let's see if I can even zoom in just a little bit. The chromosomes will condense into those structures that you're familiar with, right? Where we have sister chromatids, and let's say these are the same from mom and dad, those will be homologous pairs. Those spindle fibers will emerge from the centrosomes. Remember those microtubule spindle fibers that we talked about? They emerge from the centrosomes. The nuclear envelope breaks down. The nuclear envelope is the membrane that covers the nucleus. Well, it breaks down because we have to get access to these condensed chromosomes. And the centrosomes will move towards opposite poles of the cell. So that's prophase. Then we talk about prometaphase. So let me just point out that all of these stages are given names, but it's not like the cell goes, okay, now I'm in prophase. All right, now I'm in prometaphase. Now I'm in No, it's not like that. It's more like if I was showing you a movie and I paused during some really epic part and I said, okay, this is the Luke, I am your father, or no, I am your father scene, right? That's a memorable scene which you can always point to, to talk to people about, but it's not like the movie just stops there for a few minutes, right? What I'm talking about is that mitosis is just an ever going on process, but we have figured out really like sort of good pictures to take in that process, give them names so we can talk about it. So when we get to what we call prometaphase, the chromosomes are continuing to condense, the kinetochores appear at the centromeres, and the mitotic spindle fibers that are the microtubules will attach to the kinetochores. Remember, the kinetochores are what holds homologous chromosomes together, whereas the centromeres hold the sister chromatids together. Okay, when we talk about metaphase, metaphase happens when we see the chromosomes that are lined up right in the middle, what we call the metaphase plate. And we'll see that each sister chromatid is attached to a spindle fiber that originates from opposite poles. So the kinetochores are where the microtubules will attach. They'll attach to each one of the sister chromatids, and they'll pull them apart at anaphase. The centromeres will split. You'll get one copy going to the left, one copy going to the right. Sister chromatids are now called chromosomes because they've been separated. So this is like a duplicated chromosome. When it's pulled apart, there's one chromosome, two chromosomes. Okay? See how the terminology can sort of get a little confusing sometimes, but I think it'll be okay. So certain spindle fibers will begin to elongate the cell. You can see that the cell is sort of getting longer. When we get to telophase, we see that the chromosomes have now arrived at opposite poles, and they begin to decondense back into that chromatin, that spindly fiber, that spindle chromatin. Then we'll see nuclear envelopes starting to form around the duplicated chromosomes. Then the mitotic spindle fiber breaks down, so those tubulin fibers that attach to make microtubules, they'll start to break down. And spindle fibers will continue to keep pushing this apart. Finally, we get to cytokinesis, where we get that cleavage furrow. Remember, these are the microfilaments, that actin filaments, that are sort of pinching the two cells apart. In animal cells, in plant cells, you'll get what is called a cell plate, which is just a precursor to a cell wall, which plant cells have that makes them different from animal cells. But basically, cytokinesis ends with the formation of two identical daughter cells. Okay, so I know that is a lot of information, but that's pretty much all we need you to know, is the very, very basics of it. If you were taking a general biology course or a cell biology course, you would know much, much more and need to know much, much more about it. So everything I just said is listed here if you want to read that, but let's keep moving. So there are, like I said, there are reasons that a cell will want to control when a cell goes into mitosis. So there are also triggers that might say stop or go in terms of you want to go into mitosis, you don't want to go into mitosis. We call these checkpoints, and a checkpoint is the point in the cell cycle at which the cycle can be signaled to move forward or stopped. Again, it's not going to be too important for you to know what each of these are, but just know that if I talked about a checkpoint, it could be a reason for the cell to stop at any point. There's a G1 checkpoint, stop at G1. You stopped at cell growth. You never got to the point of synthesis. And there's also the G2 checkpoint, where you could have synthesized the DNA, you've got a double copy, you're even starting to copy other organelles, but then the G2 checkpoint might tell you you've done something incorrectly, stop. So that's all I really need you to know for that. Here are some real-world maybe implications of when the cell cycle is out of control, and those are cancer or tumors. Basically what happens is the cell loses its ability to tell this cell to stop growing, to stop going through mitosis. The abnormal cells will just continue to divide, and they'll divide and divide and divide, and that can actually damage the tissues around them, even spread to other parts of the body, which we call metastasizing, and eventually, obviously, we know can result in death. In healthy cells, we know that the tight regulation of these mechanisms prevent this from happening. But when failures happen, it can cause, unfortunately, unwanted and excessive cell division. So it's just real-world implications of when that cell cycle control fails. If you think about it, it's really, really impressive how good cells are of controlling this, right? Because this happens millions upon billions of times every day in your body. And most 99.9% of the time, everything goes according to plan. So I'm going to leave it at that because honestly, like I said, we could just keep going with this and I could go into much more detail, but that's not what this class is really about. So I'm going to leave it there. Any questions, make sure you get in touch.